me a little. There we go. That's what I was asking for. We're good to go. All right. Afternoon, all. Thanks for joining. Let's get the share going. All right. Good, good, good. There we go. Welcome, everyone. As mentioned, deeper differential expression analysis with shrinkage correction. Lots of good topics, some more technical, plenty of applied. As a quick preview for those who want to follow along on your own systems with the code examples, I'm going to be blowing this up in just a moment. But everything I'm referencing, including this slide deck, and again, all the code, a little bit of data, is available at my GitHub, Differential Expression Training, July 25. Let's see. Which is right here. And for those not versed in the alchemy of GitHub, easiest way to get these materials is just go to the code button, drop down, download zip, and that will pull everything presumably to your downloads folder. And you can open it and run as makes sense for you. All right, and we'll be switching back and forth between, oh dear. Let's go like that. There we go. There we go. Yeah. So just to make sure everyone's set up properly, I think all of this was sent beforehand, but for now or future, if you want to run along with the code, of course, we need RStudio, and this set of R packages, and there was a minor update to, in particular, DESeq2 since the last time I ran this, and in particular, sparse matrix stats is now going to be needed for the single cell portion of this section. So that's a new package that we're going to need in case you've downloaded everything else. And I'll keep, for anyone joining later, the GitHub link at the bottom if you want to go pull that data later. All right. It would be remiss of me to not point out some of the other excellent written resources on these various topics. There's lots. Here's three that I particularly like. We will, of course, be focusing on applications to DESeq2 today. And the vignette from the original developers, Mike Love, Anders and Huber, is at this link. It's fantastic, very detailed. We're going to be following some of the examples there, but talking about it in more depth. For single cell pipelines in general, the OSCA book is very good, published by various greats in the bioconductor group. And we also have for basic RNAseq a carpentry workshop, also from Bioconductor, at that third link. And before we get going, probably this is ground rules, but any questions, please feel free to add them to the chat. For me, feel free to unmute and ask questions, particularly helpful if you say start with the chat, I answer a question, and then this follow-up. Unmuting is great. All right. Let's start with looking at the data structures we're going through. I think that Erica gave a previous talk on GLMs, but I don't know if everyone was able to make it to her session, so I'm going to be doing some review of that and looking at the specific data structures that we'll be talking about. So we're assuming something that looks like this. A G by J matrix of some kind of abundance measures. We're using the usual bioinformatics format where our features for RNA expression, that's going to be genes, are on the rows, G many features, and our samples, that could be bulk samples, it could be single cells, it could be spatial spots for a spatial assay. There's going to be J many of those on the columns. And this particular data is the first couple lines from our first test data set. That's from the Pacilla gene knockdown in Drosophila test data that's available not with DESeq, but used in their vignette. And we can see the characteristic structure of this kind of data. It's integer, it's non-negative, there's a decent number of zeros, although also some features with expression levels in the thousands. And this is the kind of data that we're going to want to work with. So, going to do our first switch over here. Whoops. Pull up those scripts. Let's just get that right now. scRNA as well. Hopefully the text is reasonably visible through the Zoom. Yell at me in chat if there's an issue. But otherwise, let's just go through the quick start, just running a basic DESeq example. So first we'll load our libraries, the data itself, Pasilla, DESeq2, some statistics and plotting packages, ggplot2 and matrix stats. Read in the data. This is grabbing the file path and the metadata file path. Actually load in those data. So now we have a count matrix, count mat. We're going to do a quick filter on this, just very crude QC, making sure that the number of non-zeros in each row, again, that's the number of non-zeros for each gene, is at least 3. There's 7 samples, 7 columns in this data set, so that's not going to reduce our data size too much. Read in the metadata. And then this is going to be important, a little bit annoying because it doesn't seem to change the actual structure of the data, but DESeq requires this. We do have to make sure that the type, the data type for both condition and type, since we'll be using those two comments, is in a factor format. So we'll just cast those to factor to be extra sure. And there we go. Looks exactly the same, but now we're confident that we are indeed dealing with, say, a factor type instead of a character type, which would throw annoying and confusing errors. We will also load, this is for a later section, but just to get ahead of things, some ZBTB3 data. This should work for everyone, but I haven't been able to test it on other systems. First line is trying to point to whatever directory this script is running in, and then pull the ZBTB3 test data. If this doesn't work, just switch out this first line to be the path to where, pardon me, to wherever you downloaded the repository. The data is in the same folder as the scripts, same folder as the slide deck. And so that should load everything. That gives us count data for ZBTB3 and metadata for ZBTB3. Print that out quickly. And this is a multi-omics assay with some RNAseq samples and some ChIPseq samples pulled from a recent paper I was part of, Paul Park Molecular Cell 2024. And this is a cleaned subset of some of that particular data for a later section. But let's just go through the quick start analysis. We want to run a DESeq analysis, want to find the differentially expressed genes. In this case, we're looking at the Pasilla dataset, and we're just going to compare our treated group versus our untreated group. So let's first, another thing that can get you, let's make sure that the row names of our metadata are going to have the same names as the columns of our count data. In this case, they have this extra FB. We're going to get rid of that with this line. Make sure that all the row names of our metadata match the columns of our count matrix. All good. And then just as an extra check, this shouldn't be necessary, but it's good practice for data sets that are larger or you're not as certain of the cleaning. This is just going to reorder the columns of the count matrix to make sure that the columns of the count matrix are the same as the rows of the metadata. Strange things can happen if they're mixed orders. And just double check that columns and rows are identical. They are. We're good to keep going. This is one of the many ways that you can set up a DESeq object. My favorite because I like to work from raw data, but you can also pull from, say, a summarized experiment structure that is very well supported. DESeq data set from matrix. Our matrix is going to be our count matrix. I'm going to, more as a reminder to myself that this is happening, I'm going to pre-round the values in the count matrix because DESeq will do that internally if you don't do it beforehand. So this is a good reminder that that's happening if, say, you have something like expected counts from RSEM, which are decimal valued. There's our count data, load in our metadata, and then following the standard R syntax, we're going to create our design for this particular experiment, modeling everything just as condition. Remember that was treated, untreated in our metadata. Great, that data set is built. Now we're going to just run DESeq on that DDS object. Nice and fast. Look at the results, and there's only one comparison here. There's two levels of one condition, named condition, and so we don't have to be too fussy about results. It's just going to pull out that comparison, condition untreated versus treated. Do take note of that. Read the headings. You might expect it to be the other way around, treated versus untreated. So check the order there. It will pick a default for you if you don't specify it otherwise. This is giving us the base average expression level for our features, the log2 fold change, untreated versus treated, standard errors, test statistics, p-values, and adjusted p-values. And then we can visualize these results with an MA plot. I routinely forget what MA stands for, but the structure is on the x-axis. For those who haven't seen this, we're looking at the average expression level of the feature. On the y-axis, the log2 scale, despite that it says log, it's log2, scale fold change between our two groups. Each point is one of our features representing one particular test that's conducted, and by default, it will threshold at an internal threshold for significance and color the significant calls in blue based on adjusted p-value. And that's your basic run of differential expression analysis with DESeq. And we're going to spend a lot of time getting into the real guts of how this works, how it can break, how you can fix that going forward. A quick note before I go back to the slide deck and we talk more about what exactly DESeq is doing so we can understand it a bit better. EdgeR also exists. It's great. I don't use it very much, but that's not because it's bad. It's just I am used to DESeq. And in my opinion, the user interface is more intuitive and more flexible. And so that's why I'm presenting it today. But if you, for whatever reason, or your collaborator likes EdgeR, EdgeR is fantastic. It does most of the same stuff. All right. All right, differential expression, back to basics. So what are we testing? Most generally, this is going back to your statistics course, if you took one, we're asking whether the effect size of some combination of covariates is going to be significantly non-zero. That is the most general case. Looking at contrasts, we'll talk about some contrasts later. But in practice, we're not looking at combinations of covariates. We're probably looking at just one covariate. Oh, I see some chat stuff. Will, if it is possible, here we are. Just make sure there's nothing. Great. Will, if you don't mind too much, could I ask that if there's something pinging in the chat that you yell in my ear?
Sure.
The chat's
Sure.
on the different page.
Sure, no, I'll in your. There is a question from Chris Lee. I'm not sure if you see this.
Yeah, I just noticed that. So I don't know the specifics. Some of this is going to depend on, say, how the internal algorithms deal with, say, a sample which is all zeros. You're going to have some kind of early termination criteria. and they may differ because if you have a sample that's all zeros, you're going to have covariates that converge to positive or negative infinity if you don't stop it early, for example. Another thing that is probably the reason that you're thinking of, Chris, is that the internal normalization defaults between DESeq2 and EdgeR are subtly different. EdgeR uses the trimmed uh, median of moments, TMM. I'm forgetting exactly what it stands for, but the TMM method for calculating normalization factors, and the DESeq2 default is median ratio. They're similar, but they will give slightly different values, and that may be the reason for the difference. There's also some particularities. I won't be getting into this today, but in how DESeq2 calculates adjusted p-values, not log2 fold changes, but adjusted p-values, which is not, say, a simple Benjamini-Hochberg correction. It's something a little bit more subtle, and that could also be leading to differences. I don't know in detail how EdgeR handles p-value adjustment. Hopefully that answers some of your question. All right. Yes. So in practice, we're probably not going to be usually looking at a combination of covariates and contrast, although I will be talking about how we can do that. But usually it's just one covariate which defines one difference between two groups. And so we want to test, is that particular coefficient in the model specification strongly non-zero after accounting, of course, for the various sources of nuisance variation. And I've mentioned this before, but I'll repeat it, that with DESeq2, ratios are measured in the log2 fold changes, although, again, log is thrown about here and there.
There's a follow-up from that on Chris, from
Yep.
Chris. Okay.
Different procedures. If you can trust the list of DEGs that are typically output, is when the list from DESeq2 and EdgeR differ. So I have not done strong comparative analyses between DESeq2 and EdgeR. I've done a lot of robustness testing on DESeq, and we'll be seeing some of those today. That's part of why I rely on it. So maybe that's a simple answer to your question of, for everyone who's maybe not looking at the chat, how you can trust the output of differentially expressed genes from DESeq2 versus EdgeR when they may differ. Part of that is going to depend on just how different they are, because both of these methods you're going to be thresholding on some acceptable false discovery rate. And so you're going to have some errors in there regardless. And marginally, if they're pretty similar, your downstream analyses, say gene set enrichment analysis, are probably going to be pretty darn comparable. Hopefully, also, if you're running robust testing, you're not publishing in isolation just the differential expression results as the conclusion. There's going to be some follow-up bench work for you or your collaborator to validate the results. Obviously, not always possible. It depends entirely on the experimental design, but where possible, I do try to push my collaborators in that direction because the model is always wrong. George Bach's little paraphrase there. Sometimes it's useful, but the model is always wrong, whichever one it might be. I don't know. I think the high level is I trust DESeq2. Take that for whatever it's worth. And I know the underlying model broadly of EdgeR very similar, and so they should give very comparable results. But if they're dramatically different, then maybe the answer is use some of the tools that we're going to look at today to try and understand why, because that might indicate which one or possibly both are misbehaving in the analysis. All right. And I'm going to keep going, but of course, feel free to add in more comments, or if you want to unmute with a follow-up, we can look at that as well. Great. DESeq2 and EdgeR, but I'm going to stop harping on edge R for the moment, is based on a negative binomial generalized linear model. Again, Erica went through some of this, but I'm going to repeat it here. And that's saying that our counts, YGJ for gene G and and sample J, are modeled as some negative binomial distribution with a mean, mu GJ, and a dispersion parameter, phi sub G. Because this is a GLM, a generalized linear model, our mean is related to a linear function of covariance through a link function. In this particular case, the default for a negative binomial and the particular implementation for DESeq is a log link function, which says log mu is linear related to our coefficients and our covariates, plus, and this is specific to DESeq2, a predefined and required offset log S sub j, that is your size factor, or alternatively called the normalization factor for sample j. And then again, dispersion parameter phi j, which is closely related to the variance. Think of this as analogous to your standard deviation for a normal distribution. And indeed, we can, if you were so inclined, write your standard simple linear regression as a normally distributed generalized linear model. That is, YGJ is modeled as the same product of coefficients and covariates plus some error term, such that your error is normally distributed. And then you would write that using the same notation as below, as YGJ is modeled as normal mu sigma. Mu is modeled as no link function here, just literally as the product of your coefficients and your covariates. This is in the field called identity link. It's the most boring possible link, but it is a link function nonetheless. And I just bring this up to draw the comparison between standard linear regression, which you might be more familiar with, and this specialized case of the negative binomial GLM, and indeed GLMs broadly. That means that when you're building your models or considering testing, you can interpret and design the model very analogously to how you might be familiar with identity link. This is the mathematical structure I'm going to be referencing. We'll see the comparable version below, where we have your various different levels of covariates and your various different coefficients for each level. And with the negative binomial, the only difference really is that we have this log link. So if we apply the log to the right-hand side by exponentiating everything, we see that mu is equal to the product of all the exponentiated terms, coefficient times covariate, ultimately multiplied by your size or scale or normalization factor. This is why, for the purposes here, as a side note, I wrote this as log of S sub j. When considering the internally calculated normalization values from DESeq2, or when supplying them externally, which we'll be looking at, we calculate the size factors on the same scale as mu, so that they are a direct multiplier of your fitted mean. And of course, this model also shows directly that we're not working on the linear scale, we're working on the multiplicative scale. And likewise, just my last note here, this also means, I think this is typical practice, but I want to emphasize it in case, that when inputting your data to DESeq2, it should be on the scale of the raw expression. Only adjustments you should generally be applying are that rounding function if, say, you're dealing with decimal valued data. And you should not be, say, scale normalizing or log normalizing for DESeq2. That's handled internally through, again, this negative binomial GLM. The reasons why we like this approach for we got a question, I see. Why does DESeq2 round the expected counts? Is this a necessary step? The reason is because, yes, it's necessary. And that has to do with the negative binomial distribution which is the underlying assumed generating distribution for the data. Unlike a normal distribution, which allows positive and negative values and is continuous, any value has a probability density. A negative binomial, which is like this to a Poisson or a binomial distribution, if you're more familiar with those, it is specifically defined only on the non-negative integers, 0 to not infinity, because that's not an integer, but arbitrarily large integers. And so that's why it does need to be rounded internally. It'll still fit, but it'll do strange things. All right, getting back to why a negative binomial GLM for these data, beyond the point that we just highlighted about usually our data is integers with some exceptions like expected counts. We are also usually interested in fold changes, which are implicitly ratios between our groups that we may be comparing. And just to write that out explicitly, we're interested in usually the log fold change, mu of group A over mu of group B, which we can rewrite as follows. Log of x bar A minus log of x bar B, statistics notation meaning the sample mean as opposed to the true mean of group A if that's not apparent. And the nice thing about negative binomial GLMs is because of the log link function specifically, they deal directly with log ratios internally. There's no need for transformation. This is what's calculated by default. And the important note is that you could try to approximate this by, say, scale normalizing your data, taking the log transform, possibly with a +1 pseudo count, that's quite common, and then taking the average of your log normalized data for group A and group B, and then calculating a t-test on that difference. Similar, but importantly, that's this thing I've noted here, not the same. Log of XA bar is not the same as the average of log XA. And so they might be similar values, but they will be different, and you will not precisely, or rather accurately, calculate the log fold change if you do this method. C, another term is negative binomial GLM parametric. I've seen concerns regarding the using negative binomial distributions with sequencing data. Would it be a concern here? I am not entirely sure how the first bit and the second bit connect. So if I don't answer your question, please feel free to follow up, of course. Negative binomial is parametric in the formal sense that it is parameterized by user-defined sets of covariates and the corresponding model fit coefficients. That is your parameterization. I don't know if that's precisely what you meant with the question, so again, please clarify if not, but it is parametric. And for applying negative binomial to sequencing data, with, again, exceptions like expected counts, which are a bit strange, all the data that I've seen and worked with that's bulk RNA, single-cell RNA, both non-UMI and UMI, ChIPseq, which you can convert to counts, some other exon levels, expression. Those are going to be naturally modeled by a distribution which is non-negative integer based. The reason why we like negative binomial as opposed to another non-negative integer distribution like Poisson is specifically that the negative binomial distribution is over-dispersed, which means you can define using that parameter phi whatever level of variance you want in the fitted distribution. Poisson is fixed. The variance is equal to the mean, and the mean is equal to the variance always. And empirically, sequencing data is over-dispersed. The variance is larger than the mean. How much depends on your particular data set, but it is almost uniformly larger. Some caveats which might get to the underlying question is if you are using something like FPKM as your measure of expression, that is pre-normalized internally. That is that FPKM. You're taking counts over a region and normalizing by both the total expression level of or total sequencing depth of your experiment and the gene length. And so if you're tempted to say calculate FBKM, I would encourage you to instead consider something like expected counts, or using quantification from, say, salmon, which will give you integer valued counts. Salmon is very good. And that might get to the issues that you were thinking of. Let me know if I didn't quite cover the details of your question. But that was the third point. You folks are getting ahead of me on the details, but I think we've covered everything. The log ratio through the negative binomial GLM is not the same as calculating a t-test on the log normalized data, although sometimes it's similar, and this might be good enough for your situation. Up to you to make that call. Usually looking with integers, increasingly so with, say, the UMI-based single cell protocols or spatial protocols, which are inherently integer based. There's no room for, say, unexpected counts there. And as just discussed, over dispersion. All right, I think we're coming up pretty close on the next practical example where we get to look at some of this in practice, but some more preliminaries before then. Normalization factors. I was just mentioning how something like, say, FPKM, or here I have as an example, RPKM, very similar, slight difference in interpretation, are various methods of normalizing. The nice thing about DESeq too is that normalization is handled by the model, which means that the model operates on the raw data. It is close to what the sequencer actually observed as possible, which tends to give you more power than if you, say, pre-correct for differences in sequencing depth or other technical variation. And the way that the model accounts for this is that size factor or scale factor or normalization factor, whatever you want to call it, S sub j for each sample j. If you're interested in the model specification, this is technically defined as a model offset, which means that there's an implicit, i.e. not calculated, coefficient beta s, which is always equal to 1. That's why it's usually not stated. That's going to be, we already saw how it incorporates into the model, and you can calculate these in many ways. You can calculate it sort of through, say, RPKM, although that's usually not going to give you separate values S sub j. More easily, you can extract an S sub j from a TPM or CPM, transcripts or counts per million, measure variations in library size, the trimmed mean of M values. There we are, the TMM that I mentioned for edge R. We will get to in a second what DESeq does, but previewing that, this is why you want to bother with normalization, and in particular, bother with normalization internal to the model, because I've already discussed you don't want to pre-normalize the data. So this is actually from the TMM paper, Robinson and Oshlock, Genome Biology 2010, figure 1. And so here they're looking at somewhat of a contrived scenario, but this type of issue can increasingly arise. And that is where they're comparing some liver bulk RNA-seq all the way back in 2010 to some kidney bulk RNA-seq. And in the top, they're looking at the distribution of fold changes kidney normalized by the total number of reads for kidney 1 compared to kidney sample 2 divided by the total number of reads for sample 2. And we have this nice, plausibly normal, certainly symmetric, about 0 distribution of fold changes, log 2 fold changes. And this looks the way you would expect. You have biological replicates, kidney 1, kidney 2. Your average fold change should be 0 on the log scale, meaning that your typical gene is not measurably different. And because of noise, you're going to have some tail on either side. The issue, and this is where normalization comes in, or bad normalization comes in, is when you compare something very different, like liver to kidney, and you just use, this is implicitly a sequencing depth-based normalization. We're dividing by the total counts n sub L for liver or n sub K for kidney. And as a preview of why we look at different techniques, I'll note that RPKM, FPKM, CPM, TPM, all those PMs methods, those are depth or library size based methods. So they're going to have the exact same characteristics as this, just at different fitted mean values. And so when you look at something that's very different liver and kidney expression levels across all the genes, you see this shift where the modal fold change in this particular case goes negative on the log2 scale. Now, depending on the particular details of your experiment, that might be expected, that might even be acceptable. But typically, what we're interested in, and this is the implicit assumption in EdgeR and DESeq2, typically what we're interested in is the smaller set of genes which are different, roughly speaking, in the proportion of expression that is allocated to those genes out of the total expression across all genes. Another way to put that, which is simpler, is you would expect that your typical gene between any two groups should have a fold change of 0. Your mode should be about 0, and you're looking at the special cases. If, and there are some experiments where this has been an issue, if you're looking at a scenario where the effect you're studying, you expect it to have a global impact on RNA expression, say globally increase or globally knock down total expression. Ignore everything I'm saying. You're not interested in relative expression levels. This is a fine result. But usually we are interested in relative expression levels where it's a small subset of genes that are changing, or at least that we're interested in between two comparisons. And so this is a problem. This gives you large numbers of false positives. And here's how they justify that in a more concrete sense. They picked out the particular set of housekeeping genes, however you choose to define that, but by definition, a set of genes which you expect to not change between your two groups, highlighted those, and we can see that the median fold change, liver versus kidney, of our housekeeping genes is less than 0. So with this particular normalization method, your housekeeping genes are changing, which is definitionally a problem. So this is why we like to, or I like to avoid the sequencing depth or library size based normalization method. TMM, which Robertson and Oshlag propose in this paper, is one way to solve that. The other is the DESeq2 default, which in their paper, they don't give it a specific name that I recall. I call it the median of ratios, very descriptive of how the actual math works. And as a side note, SCRAN from Lund et al. 2016, which I'm going to be recommending for calculating normalization factors for single cell data or other very sparse data, is conceptually the same as median of ratios with some added algorithmic tweaks to deal with the sparsity issues of that kind of data. And the broad idea of the median of ratios, MR approach to calculate your size factors SJ hat just meaning your calculated estimate, is to, for each sample J, cell or batch or what have you, look at each gene G, your observed counts, YGJ hat, and compare it to some globally calculated pseudo expression level. This formulation on the bottom, that is the geometric mean across samples for gene J. Think of it as a reference expression level for gene G. You're going to look at that ratio, and in fact, you're going to look at all the ratios over genes G and pull out the median thereof, median of ratios. To rephrase this in less mathematical language, the biological assumption here, underline this particular mathematical formulation. The biological assumption is that your median gene between any two groups should have a fold change of 0. And so this is calculating size factors Sj, which should cause that to happen. And so that's the idea behind median ratio. TMM does something very similar. All right, a little bit of a note on SCRAN. I already mentioned does the same thing. It gets around the zeros. You might notice if you're looking at the details of the math, we've got a log in here of YGJ for sparse data like single cell or a bunch of the spatial variants. YGJ might be 0. log of 0 is a problem. And so this doesn't work for sparse data or in general genes with any 0 in that gene. And so Scran gets around this by pooling cells together and then a more complicated deconvolution method. All right, enough background, GLMs, size factors, ratios. Let's go back to the analysis script and look at some examples, unless there's any last minute questions before we do that. Type those if you have them while I'm pulling up the code. All right, going to look at normalization factors, their impact on the model, and how you can use custom ones if you are so inclined. So first, let's calculate some custom normalization factors. We have reads per million space size factor, not reads per kilobase million. I'm not doing a gene correction, just reads per million. And that's the simplest possible thing. Total number of observed reads per sample, that's your call sums of the count matrix, divided by a million. Reads per million, it's self-explanatory. And we can calculate the normalized matrix, norm at RPM, by just dividing each column by its corresponding reads per million factor. Let's just print those out so we can see them. This is what those normalization factors look like. They are comparatively large in the scale of size factors, meaning greater than 1, just because these are deep sequencing. We have many more than 1 million reads per sample. The method for calculating the median ratios manually is a little bit more involved. I'm going to start by doing a crude filtering, make sure that the row sums of the count matrix that are less than 5 is 0. So that's just saying make sure that we're only looking at the genes where all samples have at least 5 counts for that gene. Just cutting off the bottom lowest expressing. That's still pretty large. We're dealing with almost 8,000 genes even after that filter. This is also getting rid of those zeros, which cause an issue if you don't handle them. Calculate your geometric mean for each gene. This is your reference expression level. And then calculate your median ratio size factor. So for each column of the data, divide your counts by that reference expression level. Level 2 for the apply, say, look at the columns of the matrix right here, and then calculate the median of the ratios, MR. And we can look at those median ratio size factors. Here they are. We'll compare them to RPM in just a moment. But first, we can calculate a normalized expression matrix, norm at MR, by likewise dividing the columns of the count matrix by the median ratio size factors. We can also go back to our quick start where we just plug the raw data into DESeq2 and pull off, using the size factors function, pull off the default calculated size factors, which I'm claiming at least are based on the median ratio method. And then let's visualize all of these. Here's our reads per million up top. Here's our median ratio factors on the bottom. And we can see that they increase linearly, roughly, with the default factors that's pulled off directly from DESeq2 on the x-axis. And in fact, the manually calculated median ratio size factors are indistinguishable from the default calculated. Any small differences between the blue dots, median ratio, and the black line, which is just a slope 1 line showing equality between calculated factor and default factor. Any variation there is just going to be due to how DESeq does its pre-filtering to figure out which genes to include and not include in the calculation. RPM follows this same slope, so they should give similar results, but just uniformly at a much higher level. Now we can do some more visualization, build just some plotting data, visualize the plot, and this is the way that I like to visualize, at least for a bulk experiment, whether the normalization is behaving itself well. And we can look here on the left, the median ratio and the reads per million size factors. This is calculated on the normalized matrices, where each box plot is one of our separate 7 samples, treated 1 through 3 or untreated 1 through 4. And the thing that I look for related to that assumption that between any two groups, your median gene should be the same after normalization, I look for whether the median from these box plots is roughly the same across samples. And we can see that for both MR and RPM, that is the case. These are plausible normalization methods for this data, despite the criticism I was previously leveling at a depth-based method. Often it does work. If your samples, your conditions are not wildly different, a depth-based normalization method is probably going to be just fine for you, as is the case here. We do see that RPM is uniformly lower. That's not surprising because we saw that the RPM size factors are uniformly higher. You're dividing by a bigger number, it should be smaller. Let's then look at how we can use these custom size factors for a DE-seq analysis. We're going to first build our RPM, same initial setup, rounded counts, metadata, model as condition. Here's the first difference. We manually set the size factors for this DDS object as RPM SF with this line. Then build the model, visualize the top results. Here they are. And then let's do the same thing for median ratio size factors. Build the model, same setup, only difference now is we apply the median ratio size factors instead. Build the model, visualize the top results. Here they are. And now let's compare them with MA plots. And these are the results of that MA. Let's zoom in a little bit on that. And first off, they look pretty darn similar, but you'll notice 2 changes, and we'll get into why and how you should think about this in the next section once we go back to the slide deck. First off, and this should be expected, the x-axis, mean of normalized counts, RPM is uniformly smaller than median ratio. That's expected. The RPM size factors are uniformly larger. It should just shift the overall average normalized level left. That's fine. That's not an issue. The weird thing is, notice how our choice of normalization method has dramatically changed the number of significance calls between RPM and median ratio. And this is even though the calculated effect sizes, look at some of our particular high fold change values. This set of 3 looks almost identical to the set of 3 over here. Effect sizes look identical visually. I can see very minor differences due to the very minor differences in size factor, but not enough to explain why we have this dramatic difference in the number of calls. Hold on to that thought. In the next section, we'll start looking at why that might be the case. All right. To get at this, now we need to look back at the negative binomial model. And the other part of it, we talked about the mean, which is how you define your covariates, define your comparisons, construct the model. Less studied, or at least less discussed, is the impact of the dispersion parameter, phi. Phi, I mentioned before, is analogous to the standard deviation in a sigma. In a normal distribution, it is related to the variance, and in particular, our expected value for a negative binomial random variable y is mu. Great, that's exactly the same as for normal. The expected value is mu. The variance, however, is not just a sigma squared, as in a normal distribution. It is the mean plus the squared mean multiplied by phi, your dispersion parameter, which is to say your variance increases proportionally to phi for any given level of the mean mu. One note on this particular formulation, it's not going to matter too much in practice, not going to matter very much at all for today, but this particular formulation is unfortunately not universally accepted. Variance is mu plus phi mu squared. At least half the time, and maybe in fact more than half the time, this is instead written as mu plus 1 over phi mu squared. And different methods that use a negative binomial method will choose one formulation versus the other. Today we're using this one for DESeq 2, which is nice. Larger phi means larger variance. But you will have, if you have reason to look at the variance estimates or the dispersion parameter estimates for whatever method you might be using, if it's based on negative binomial, you'll have to check, unfortunately, which formulation they're using because it's, again, not uniform. All right. Yes. So one challenge here is we don't just need to fit mu, as in standard linear regression. We also need to fit phi. One of the issues for sequencing in general, not specific to, say, the negative binomial model, which we're using, just sequencing in general, is you have many features you want to test, comparatively few samples, sometimes, maybe often, very few samples, like we saw just 7 between two conditions for the Pasilla data set, which means you're going to have a high standard error on your fitted phi, and that means phi is going to be typically overestimated, and that's going to increase corresponding to the standard error on your fitted coefficients, which is going to, in turn, reduce your power to identify your significant effect sizes. So one way around this, which is implemented specifically in the DESeq method, is to implicitly pool information across similar genes. Similar here is defined as across all your samples having a similar expression level. And the idea here is if your average expression between two genes is about the same, your variance is probably about the same. Certainly, the mu squared terms are going to be identical, and we're just going to assume that phi is roughly constant given a level mu. That doesn't have to be true, but empirically it seems to work pretty well. And as a note, before I move on, the MLE, the maximum likelihood estimate of phi, only exists when the sample variance is greater than the sample mean. This is one of the differences between the negative binomial and the Poisson. Poisson, no dispersion parameter, variance is the mean is the variance. Here, this is only estimable, at least via an MLE, if the sample variance is larger than the sample mean. This will come back as something we take note of later. All right, enough of the theory there. Let's go back to the script and play with dispersion parameters. All right, exploring dispersion parameters. Let's actually visualize them. DESeq 2 makes this very easy, and let's look at the dispersion parameter estimates for median ratio and RPM. Great, that comes just from the plot dispests. Plot dispersion estimates. We get to see, pull this up a little bit larger. We get to see for each normalization method 3 different terms: the per gene estimated dispersion parameter, log scale on the y against log scale average normalized counts on the x. The fitted value, this is an internally calculated parametric curve fitted to the black counts. And then the final dispersion estimate for each gene. Each gene does get its own unique dispersion parameter. And the way DESeq handles this is using a Bayesian model around the fitted values. It shrinks the calculated gene estimate dispersion parameters towards that fitted value with 2 caveats. First, the stuff at the bottom, the stuff that looks like 0, it's set at 1e-8. These are the genes where the sample variance is less than the sample mean, and so the dispersion parameter collapsed towards 0 and was not estimable. And they just got stuck at the bottom of the plot here. They get somewhat ignored and thrown up, I believe exactly to the fitted value. And the other bit, this is a nice thing to protect you against false positives, is DESeq will do some internal checks for individual genes with particularly high calculated dispersion parameters. These are genes with much more sample variance than you might expect from the normal variation around the fitted line. And it does not shrink those values. It preserves them to, again, protect you from the possibility of false positives on those genes. The main thing, though, is that the median ratio dispersion estimates are very different from the RPM dispersion estimates. We've got this uniform shift up. In RPM, we've got this capping. At 1e-1, we have this kind of visual capping line lower bound. Here, something very strange is going on. And this should be somewhat surprising, actually, because the dispersion parameters are calculated not based on the normalized expression, even though that's how we're plotting them. Recall that the variance from the slide is the actual fitted mean, mu, which accounts for size factor. Remember that log-link function. It's based on mu plus phi mu squared. And mu, and naturally mu squared, should be the same between median ratio and RPM. Because remember that they both followed very closely that slope one line. RPM was, yes, uniformly higher by roughly a factor of 10. RPM are about 10 times the median ratio size factors. All that means, though, is that your fitted intercepts, which is implicitly what we're plotting on the x-axis, should just be divided by a factor of 10, and your fitted means per gene per sample should be about the same. But obviously that's not the case. Let's go then look at this plot. We get a clue of what's going on from that x label mean of normalized counts. Let's visualize the next exploratory plot. So here we're looking at the mean variance relationships between our three versions of the data, either unnormalized, just calculate the row variance on the y, the row mean on the x, unnormalized data, median ratio normalized data, RPM normalized data. First off, unnormalized versus median ratio, we can see again why we want to use normalization factors. We have not really shifted the data left or right in any meaningful way. The x values for each gene in unnormalized and median ratio are about the same. However, the gene per gene variance has uniformly decreased pretty much for the median ratio normalized data. So that is removal of nuisance variation, which would otherwise make it hard to call significant results. Not quite so for the RPM values that we should expect this. The scale of the factors are about 10. I do see the question. I'll get to it in just a moment. The RPM values are, of course, about scale factor of 10 larger, so they shift left. But there's also something strange going on if we look at the data a little bit differently. And let's see, we have a question. Shrunken values seem generally to land more often lower the line than above it. General pattern. Yes. Previous here tend to fit more below than above. That's going to depend a little bit, of course, on the specific Bayesian shrinkage parameterization that they're doing and whatever internal sanity checks are going on. But I believe that the simple answer to that is just that the fitted value gets pulled up a little bit because you have this top tail for these particularly high dispersion values. Note that this is on the log scale, so they have a lot of pull compared to the ones below. And so that's pulling the fitted values a little bit higher than you might expect. And conversely, being based on more sparse data, the lower fitted dispersion parameters are going to be shrunk more by the Bayesian shrinkage. And so I think you're just getting sort of a little bit of an artifact of the log scaling and the information levels of high dispersion versus low dispersion, which is causing that kind of bunching below the line for the fitted values compared to above. Good question. All right, let's look at the next mean variance plot. Exactly the same data, gene variance on the y, gene mean on the x, but I didn't tell you this. I scaled the x and the y based on the square root transformation. The actual counts are exactly as the labels indicate, but now let's do a log transformation instead because that's going to reveal something different. Exactly the same data, now visualized on a different scaling of the x and y axes, a log scale instead. And I want to point out the RPM. We've just taken a massive chunk by using this RPM scaling, taken a massive chunk of the genes, the features, and pushed them below the black curve. The black solid line is the mean equals variance line. Implicitly, phi equals 0. Dashed line and dotted line are increasing, respectively, estimates of phi, what that particular mean variance relationship would look like. And recall that for a negative binomial, if your sample mean is less than your sample variance, sorry, other way around, sample variance is less than your sample mean, you cannot estimate phi at least via a maximum likelihood estimate. And so all of these genes just get thrown out of the estimation. We can actually see that even more explicitly if we narrow in just on that RPM value. This is almost the same thing. We're looking at log 1p gene means on the x-axis, and now we're looking at the difference between the gene variance and the gene mean. And we see all of these features below that 0 line. All of this gets thrown out of the estimation procedure. Why is this? Well, that's because, as you may remember from your stats class, if you took one, that if you take a random variable and you scale it by some constant, here we're scaling our random variable, which is our counts, by our normalization factor, then the variance gets scaled by the square of your scaling constant. If we divide by a size factor, then the variance decreases by the square of that size factor, whereas the mean only decreases proportional to the size factor. And so that's how this is happening. That's how our sample variance is dropping below our sample mean. And also, it appears, I'm imputing this because from behavior, I've not actually been able to ask Mike Love about this. It appears that the way DESeq is calculating dispersion parameters is a method of moments estimate or something similar based on the scale normalized data rather than, say, an iterative back and forth procedure between fitting the means, fitting dispersion, fitting the means, fitting dispersion, not least because that would take forever if you were to do it. And so when DESeq is calculating purging dispersion parameters, all of this gets thrown out. Another question. Is there a relevant situation where we want a distribution that works with data that has lower variance than mean? I have not seen that for sequencing data. That could theoretically happen. But if that were the case, curious question, but the answer might not be relevant because, again, I have not seen that in a practical example. If that were to happen, I would suspect that you're dealing with data where you are not lower bounded. This is obviously just a guess. It's a hypothetical. But I would guess in that kind of scenario, you're not dealing with data which is lower bounded by 0 and integer value, or maybe it is integer valued, but your observed counts are on the scales of at least tens, probably hundreds or thousands. In that case, I would suggest maybe just using a t-test. Again, issues around if you want to directly calculate fold changes and mean of log versus log of mean that we talked about before. You could explore various, say, negative normally distributed GLMs if you wanted to do that. The nice thing there, though, the point of using a normal distribution is you can set or calculate empirically your sigma, your standard deviation, to be anything, meaning arbitrarily small. That's how you'd recover the power in that situation. But to the core question, I can't think of a relevant situation where that occurs. All right. Yes. All right. So let's fix this issue with RPM. Let's say for whatever reason of biological assumptions or crotchety collaborators that we really, really want to use RPM instead of uh, median ratio. I don't actually mean that about the crotchety collaborators. Everyone I've worked with has been fantastic. But let's fix RPM. And the way we fix RPM is by not dividing the counts by so much. Let's just scale the RPM. Let's take RPM and divide all of our RPM values by some constant value. In particular, let's take the geometric mean of the RPM and let's calculate RPM scale. This is what the values of RPM scale look like. And notice that, visually you can't notice that the ratios are the same as RPM, although they are because we just divided everything by a constant. But the important bit is now they're centered around 1. And now we can look at scale norm, which is scaled RPM, calculate the manual size factor version using scaled RPM. Here it is. Calculate the results, plot the dispersion estimates, visualize the top hits. And this is the key bit, still using RPM-based size factors, but now they're centered around 1 multiplicatively. And we see fitted dispersion parameters that look visually identical to default median ratio or manual median ratio. And so the takeaway of that whole long spiel about size factors and dispersion parameters is if you're going to use manually calculated size factors, make sure that they're close to 1. And that's easy to do by just dividing them by the arithmetic mean, or better yet, the geometric mean of the counts. And there you go, you're done. Yes, we have something new here from the last time when I did this talk. So this is getting now to the ZBTB3 data. So this is a matched RNAseq and ChIPseq data. Again, I mentioned this is a clean subset of the data from a recently published paper. And this is where we're introducing, let's just print the metadata so we can see that. We're looking at 2 different genotypes, P401, P402, which have different mutations. P401 is a non-functional mutation, P402 is a functional mutation, and that functional mutation is altering the binding structure of the ZBTB3 transcription factor. That means that we expect the ChIPseq binding of ZBTB3 to be altered in P402 versus P401. And we suspect, and we'd like to test if this is the case, that the RNA-seq expression, when we either induce through a DOX-inducible expression, if we induce that mutant form of ZBTB3 in P401 versus P402 versus DMSO, just the RNA-seq control for each genotype, we suspect and we'd like to test whether the gene expression changes corresponding to the ChIPseq change. So let's do something a bit basic, a bit naive. There's your foreshadowing. And let's just take all the data. As a sidebar for those who are more familiar with ChIPseq, I have pulled for a subset of genes that have both ChIPseq and RNA-seq data. I've pulled the ChIPseq peaks from the promoter region of the genes. That's the per gene level expression for the ChIPseq data that we're looking at here. Let's build a DE-seq data model on all our count data for ZBTB3, RNA, and ChIP together, the metadata that we just looked at, and model it as treatment. And now I'm going to start introducing this -1 notation. I really like this. It just saves my brain when I'm dealing with more complicated comparisons. And then the 1 condition 2 levels of the previous example, this says, don't fit an intercept to the model if you're not familiar. Just treat every different level of treatment, and we have 6 of them, as its own separate mean and fit it separately, and then we can deal with what we want to test against what after the fact. Model fits exactly the same, but testing and interpretation are simpler in my mind. All right, we are going to use manual size factors here. The internal calculations for the RNAseq would be fine. There's nothing weird going on here, but ChIPseq normalization is a bit more challenging. Fortunately for us, in the metadata, we have separately calculated size factors, in particular for the ChIPseq data, and so we're going to use those. Build the model, plot the dispersion estimates just so we can see them. Here they are. Nothing particularly wild. That looks like a very similar pattern to the Bacilla data we were looking at before, with just differences in the overall scale. And then let's do 2 tests using results. I'll get into the details of formatting results a little bit later when we look at shrinkage. But plot MA comparing docs P402 versus docs P401. So treated in both cases, but functional versus non-functional mutation. And likewise, functional versus non-functional mutation for the ChIPseq data. And zoom in on that. This looks like maybe it's fine. Nothing particularly strange here. There's maybe a bit of excess in the called significant genes in the RNA-seq, but this is the results that we get. Let's dig into this though to see if it makes sense if we actually should trust these particular calls. Foreshadowing, we probably shouldn't. But let's break that down. Let's build a separate model for just the RNA-seq portion of the data. Subset the columns for just method equals RNA-seq. Build that. Likewise, build a separate model for just those columns, method equals ChIPseq. And now let's compare the dispersion parameter estimates across all 3 models. Merged, RNA and ChIP together, just RNA, just ChIP. And here we see something a bit curious and maybe should be a bit concerning. Here's the merged model. It looked fine before, but if we look at the calculated dispersion parameters for the RNA-seq by itself, these are, especially for the higher expressing genes, lower. Not for the lower expressing genes, you've got a steeper negative slope here, but for the higher expressing genes where we expect to see the most significant change, lower dispersion parameter, meaning less variance than in the merged model. But much more importantly, for the ChIPseq data by itself, much higher dispersion parameters across the board. We don't get much below 1e negative, whatever that is, right in between 1e negative 2, 1e negative 1, whereas both in the merge we're below 1e negative 2, and at the high end of the RNA only we're below 1e negative 3. So much greater variance in the ChIPseq only data. What does this mean for our results? Well, that means that we are calculating, if we trust the result, and I'm going to suggest that we should, we're calculating significance values for the ChIPseq to ChIPseq comparison using a dispersion parameter on the left for the merged model, which is too small, meaning we're modeling the variance to be too small, which means that our standard errors are too small, and so our test statistics are too big, or in other words, too significant. Summarize all of that down to one phrase. We should expect that our merge analysis of the ChIPseq data to be giving us false positives. Let's fix that. In this particular case, it's pretty darn easy, because at least for the analysis that I was doing for this project, and I suspect this is the case for most such analogous analyses, we are not actually interested in comparing the RNA-seq expression directly to the ChIPseq expression, or pick your multimodal comparison of choice. Rather, it is more likely going to be the case that we want to separately calculate our effect size in one mode, then calculate our effect size in the other mode, and then ask as a downstream meta-analysis, how do these effect sizes relate to each other? Do things go up at the same time? Do things go down at the same time? Maybe a gene set enrichment analysis type of deal. The point for us right now is we test within modality, so let's build the model within modality as well. So that's exactly what we're going to do here. Just build that whole big block and then blow it up. So on the top, we have, for just visual recollection, the merged results, RNA and ChIP together. And at the bottom, we have our MA plot for RNAseq when we modeled RNAseq data by itself. And we see we actually get some more values calling it significant if we trust the model, and I think we should. This is suggesting that we have improved the power of the test by shrinking down that dispersion parameter closer to what is suggested by the data. But much more importantly, look at the difference in the ChIPseq data. First of all, you've got a big difference in the X values, mean and normalized counts. That's not too meaningful. That's just because the overall expression levels of the ChIPseq per gene are different than the expression levels of the RNA. And the RNA counts, just because there were more samples, dominated the mean of normalized counts measure for the merged data set. So you just look at a very different subset of the data, you get different mean normalized counts. That's not surprising. But much more importantly, we get this much different distribution of log fold changes, and in particular, they look much more unidirectional, which for this particular experiment happened to be more expected. The P402 mutation altered the way that ZBTB binds actually to itself, forming these long polymer chains. We disrupted that. You no longer have polymer chains, which are endogenous in cells, and we expected that to change, and in this case, degrade the binding rate with DNA. So this is more in line with what we expected, and now I'm more trusting of the result, given that we've better modeled the dispersion parameter. All right. About to go back to the slide deck. Take a moment to post any questions if we have them before we transition to a new topic. Or just unmute and ask them out loud if you're feeling bold.
Hey, Jared.
Yeah,
Quick question. I'm not sure how you're feeling about being on time. I think you have plenty of time left, but I didn't know if you thought you had time for everyone to take a break and maybe stretch their legs, refill their coffee, anything
absolutely.
like
I
that.
was definitely thinking about when we might want to do that. And
It seems like you had a break, but I don't know your slides, so
yeah, no, we've got a couple break points. We're approaching halfway through time, so yeah, let's maybe take 5. Thanks for bringing that up. So
Uh, so it's 2:23. So let's go ahead and reconvene at 2:28 for people, if that's okay.
sounds good to me. Well, we're at time. I'm good to go. Will, do you want to give the thumbs up? Fantastic. Let's jump in.
Looks like there was a
And
question in the chat, by the way.
yeah. Next question. Yeah, there was a question about TPM and RPM. We got one response from Chris, which I partially agree with and partially disagree with. Of course, details will depend on your use case and how the data actually look. But remember that both TPM and RPKM are per million reads. So one simple thing you can do is just literally multiply no, sorry. Can't literally multiply by a million. But what you can do is simply round and then maybe do a somewhat more stringent filter on the lower bound because your values are going to be proportional roughly to the original reads. And if the counts are high enough, then you're not dealing with the highly discrete nature of the negative binomial around 0. And if you're dealing with TPM or RPKM, it's probably bulk, and you probably have high enough genes reads, at least for the genes you care about. So you could just round, do a more stringent filter, and throw it into DESeq. Or if you're just, say, interested, this is an alternate, depending on your use case, if you're more interested in, say, the direction of change more than calculating precisely the log fold change, you could take your TPM or RPKM values and honestly just throw them into a t-test, use a standard linear model. It's not going to be as precise as raw counts into DESeq, but for your use case, it might be good enough. Just raw counts, standard linear model will give you direction appropriately, if not log fold changes, or you could approximate that with the log transformed again, then standard linear regression options. None of them ideal, but you're in a non-ideal situation with TPM, RPKM only. All right. Let's keep going. All right. One of the beauties of a system like DESeq2 or any generalized linear model, but DESeq2 makes it relatively easy, is, of course, the option for multiple covariates, multiple covariate levels, and or multiple different covariates, or within a covariate, multiple levels. Because you can use just sort of your standard linear regression model design to encode that more complex design matrix. Typically, for these kinds of analyses, we're dealing with factor level covariates, meaning each covariate encodes a group membership, although you can also have a continuous covariate. Implicitly, our size factors, our normalization offsets, are a continuous covariate, albeit one with a fixed coefficient. But you could imagine others for, say, various continuously valued phenotypes of your data. But usually we're dealing with factors. So these will be encoded as a demi-variable format. That means it's just the covariate gets turned into a 1 or a 0, depending on whether that particular sample is a member of the group or is not a member of the group. And so if you have a 3-group model, 1 covariate, 3 levels, you will have an overall intercept, BG1. You will have dummy variable BG2, which is the change in expression if xj is a member of group 2, and BG3, the change relative to BG1 of your expression if xj is instead a member of group 3, or in the particular notation I used here, group A, group B, group C. This is how those coefficients reduce to the overall log mean. We can generalize this to more complex comparisons where we want to do not just a simple difference in means, but something more nuanced. We'll see an example of that in just a second. Or where we have more covariates than just one multi-level treatment group, say. In the most general case, we reduce this to adding up some collection of covariates beta k in group C1, subtracting off some other group of covariates beta k in group C2. Group C1 and group C2 do not overlap. There's no betas that are in both groups. Calculate that difference of sums as a test statistic C, and ask the statistical question: is C significantly non-zero? Null hypothesis, C is zero. Alternate hypothesis, C is non-zero. We have something directly equivalent for the negative binomial GLM log link function, except now instead of testing for linear differences in mean, as in our standard simple linear regression, we're looking implicitly at fold changes, ratios between our covariates. So we would be testing, say, is mean of gene G in group B over mu of GC. Is that significantly different from 1 or not? And this reduces, or rather expands, to using the coefficients from before, groups A, B, and C, to e to the beta 1 plus beta 2, e to the beta 1 plus beta 3. Those are the sums that define the log valued mean in group B or group C, respectively. We can algebraically reduce this ratio to a single exponential. Then remember that do some canceling of beta 1, we get just beta 2 minus beta 3. And then remember that we're dealing with a log link function, so we can just take the log of this, and our C is going to be beta 2 minus beta 3. Ask the statistical question: is this difference significantly different from 0? And recall that this difference represents the log ratio between group B and group C, which is what we were interested in. Note that because this is how it's formally defined, I have written everything in terms of natural logs and exponentials in DESeq. I've said this before, but I just want to remind us all, DESeq 2 implicitly uses log-based 2. So it's going to calculate something very similar, but slightly different from my exponential beta 2 minus beta 3, which gives the log ratio. The coefficients will be ever so slightly different for DESeq 2 because it'll give you the log 2 ratio. Here's a real world, because I'm going to show you the example in the ZBTB3 data, example of a more complex relationship. Here, we want a ratio of ratios. And this can arise where you have, say, you want to test the effect size of a treatment, and you have 2 treatments, fine so far, but each treatment is applied to a different population. Or more simply, same treatment, you want to calculate effect size of that treatment across 2 different populations, look at population-specific response. And so you would say, look at the mean of treated versus untreated in population A, and the mean of treated versus untreated in population B, and you want to know, is the response in A meaningfully different than the response in B, separate from the question of, is there a response overall? So that's your ratio of ratios. Hard to do this robustly with simple linear regression. It's even worse than the log of logs, or taking the logs of your data situation. But very straightforward for the log link GLM. Just expand this out. There's a little more fractions, but we reduce to a ratio of exponentials, 1 exponential. In this case, there's no canceling because we have all 4 different coefficients, and then convert that to log R, our actual thing we want to test whether this is different from 0, as this sum of terms minus a different sum of terms to directly relate to this ratio of ratios test. So in DESeq 2, it's relatively simple to test arbitrary comparisons. You just have to figure out which coefficients should go in the first group that gets summed and which coefficients should go in the second group that gets summed. That's some algebra by hand, most likely. Let's actually look at that in practice for the two example data sets we have. Nope, not there. Here we are. Back to the Bacilla data first. Let's look at our model matrix. This is our design matrix, if you prefer. This is currently relatively simple. One covariate, two levels, and so we have a global intercept, which defines the mean expression level for the treated group. And then you have a second covariate, which is your dummy variable, defining the difference in expression for untreated versus treated. If your sample is in the treated group, then you get a coefficient of 0. Otherwise, you get, rather, covariate of 0. Otherwise, you get a covariate of 1 for untreated. We can recall the results from our original Bacilla data. This is, I believe, exactly the same as we calculated at the very top. Just remind ourselves of what it looks like. You pull off results from a DESeq data set just by calling the results function on it. Here again, one covariate, two conditions. You don't need to do any fancy specification to get the result. But to show how we would do that for a more complex scenario, let's build the same simple model. Here, it's the same type of model as our DDS quick start, but notice the one difference I've made is now it's condition -1. We're no longer in the situation of the, say, model matrix here, where our intercept is a full column of ones. The implicit model matrix for this DDS simple is 3 ones in the first column, defining the mean for the treated group, then zeros. And then the second column stays the same, but now it's defining the global mean for the untreated group as opposed to the difference in mean for untreated versus treated. It just makes life simpler because if we go here, print out results names, this is very helpful for coding up a more complex results call. I see the two different levels in my data, condition treated or condition untreated. You can look at the man page if you like. For everything you can do with the results function, I'm going to highlight first contrast here, and contrast can call a few different types of inputs. Somewhat confusingly, you can give it 3 different kinds of data, and I'm going to show all 3 to give the same type of comparison. The first thing you can give contrast is a character vector of 3 values. The first value defines the name of the covariate that you want to compare, and that's condition. We only have one covariate in this particular data set, and that one covariate is condition. Notice it's the start of the results names for each group condition. The next value is the numerator of your ratio, the thing you want to compare. We want to compare a treated group, and then last, the thing you want to compare your target group against. And these should be different levels of your covariate. Treated and untreated are 2 different levels of condition. So let's look at the top there, and let's actually, just for comparison, recall this one. So on the top, we have our new call simple res1, where we manually define the comparison, and below we have the default. And if you look at the p-values, all this p-adjusted, all the same, p-values, all the same, log fold change standard errors, all the same, means, all the same. And the difference is your log2 fold change and your test statistics. And these are almost the same, but notice the difference in sign. And that's because by default, in the quick start, without specifying anything, the model compared untreated versus treated. And in the manual version we just did, we switched that. We did treated versus untreated. And so you just have a sign change in the log2 fold change. Everything else is identical. All right. We can also, and this is the version that I like because it's very explicit about what is being compared, how it's being compared, and it's the most fully flexible for the complex, say, ratio of ratios model. That's simple res2. Here we're doing the second type of thing we can give to contrast, and that is a list of 2 character vectors. You do have to be careful. You do need the C for concatenate on both of these. If you just do list without the C of 2 levels, it's going to throw an error. And if you're like me, it'll take you 20 minutes to figure out why, and then you'll feel silly. Not speaking from experience at all. And that's the main thing. And do see the question. We'll get to that in just one moment. And then the first term, character vector, here there's just one element, but generally it is a character vector of all the terms that you can see from results names. It's the full results names object, so condition treated, but all the terms, all the coefficients you want to add up in the numerator over all the terms you want to add up in the denominator. And if I remember correctly, I haven't done this for a while, but if I remember correctly, this is robust to redundancy. If you remember in the example in the slide deck, we had the comparison where covariates B1 canceled for the dummy variable with 3 levels example. If you have, I believe this is the case, if you just have copies of a covariate in the numerator and the denominator, either it'll run fine and give you the exact same result, or it'll give you a warning which is clear, just says don't have the same covariate in the numerator and the denominator. Either way, it's relatively simple to fix. Let's just throw this out, and exactly the same as simple res 1, just a different way of defining the comparison. Then we have a question: why remove the intercept if the results stay the same minus the sign change? You don't have to. You absolutely do not have to remove the intercept. I find it easier to define comparisons this way. If you do not remove the intercept, then there is going to be a default condition which everything else gets compared to. And especially if you have multiple covariates, not the situation we have here, but in many scenarios, you may have multiple covariates, each with multiple levels, then figuring out the algebra of which coefficients need to get added in the numerator and which coefficients need to get added in the denominator, recalling that everything is relative to the default, that can be more convoluted. Mathematically identical, but unnecessarily troublesome. Another question, genes with a p-value, but missing adjusted p-value, adjusted NA. Yeah, I've read that this may occur due to independent filtering. Exactly correct. When the multiple testing correction is applied to genes with valid p-values. That is very deep in the weeds. This is DESeq. I'm not going to get too much into the math of what's going on. It's a bit involved, but this is DESeq actually trying to do you a favor, even though it might seem like it's not. The statistics is reasonably robust, and DESeq is looking at, and by the way, this shouldn't happen to a significant p-value. It's generally going to happen to your naturally non-significant p-values. And what's going on is DESeq, with this independent filtering that's mentioned, is looking at all the significance values and doing something a bit more sophisticated than, say, just a Benjamin-Hochberg correction, and trying to parse with extra data what subset of p-values are most representative, roughly speaking, of true effects, and calculate adjusted p-values just from that set. And so everything that gets tossed out of that set gets flagged by this NA in the p-adjusted value. If you don't trust this independent filtering, for whatever reason, totally valid. I didn't exactly go into depth of how it's working. Or you just want to get adjusted p-values for everything. It's very simple. Just pull off the p-value column, apply a Benjamin-Hochberg correction to it directly. Or I would suggest instead do what we're going to walk through in a later section and apply posterior shrinkage and calculate, instead of a p-adjusted value, calculate an S value, and that should give you significance measures for all your genes. No independent filtering involved. And we have one more question. When would we want to not remove the intercept? So for that, when would we not want to remove the intercept? I almost always do, but if you have a very simple design, one condition, two treatments, two different levels, and you don't really care about the sign or you're going to check it after the fact, just do the simple model without the -1, pull off results, and just check sign based on what gets compared to what. As mentioned, the results don't change. It's just whatever is most convenient for you. Finally, going back to the things that we can send to contrast in call-in results. I think I've done this once to suggest how often it gets done, but this is actually the most powerful version of applying contrast. If you took formal stats, this is the formal definition of a contrast. And this lets you do not just sums of coefficients, but linear combinations of coefficients. That means instead of, say, adding all your coefficients in one group, you can do a weighted sum of coefficients in your numerator group, a weighted sum, you choose the weights in your denominator group, add those together. And then this vector C, which should have length equal to the total number of coefficients you have, not just the length of the coefficients you want to compare, is going to give those weights for every single coefficient. It should be 0 for coefficients you want to leave out. And if I remember correctly, you want it to sum to 0, as you see here. This is just the most simple possible one. Weight of +1 to condition treated. That's our first covariate. Weight of -1 for condition untreated, meaning subtract that one. And this is just a different way of doing that linear comparison. And we can see, again, exactly the same results as before. I almost never do this, but it has happened once or twice when, again, you need, for whatever particular reason of your scenario, you need a weighted sum instead of a more typical unweighted sum. And then lastly, we can do, say, a full model matrix here. We're going to have condition and type, because this Bacilla data included both treated, untreated, and single end and paired end reads. And so we can correct for, say, the biases introduced by single or paired end reads, respectively. You decide which one you think is biased relative to the other by accounting for that in a separate variable. And this is particularly where that -1, in my opinion, starts to become handy. You can keep exactly the same contrast format, but now type is naturally corrected for in the linear model. And you get the same kind of results, but now different values because you've corrected for difference in sequencing type. Similarly, you can pull off just the results of type single read. This is going to be the comparison of single versus paired. This is the effect just of being a single end compared to the default of being paired end. This is one annoying thing. The -1 only gets you so far if you have multiple covariates, not just multiple levels, but multiple covariates overall. Some of them are still going to be against a reference default, but it's a little simpler. But this gives you the effect sizes of single versus paired end as the implicit default. Let's do something similar very quick before we move on to the next section. For the ZBTB, that's a typo. ZBTB3, definitely not ZBTB4, but you get the idea. Let's pull off the different conditions for just the RNA component of that model. And we have 4 of them. And previously, we looked at the treated, that's doxycycline, the treated comparison between functional and non-functional mutation, P402 versus P401. For this particular analysis, what we were actually interested in was that relative response relationship that I was mentioning in the slide deck. We wanted to know under treatment with doxycycline, did the RNA change more or broadly differently in P402 than it did under treatment in P401. And so we do that ratio of ratios comparison. Here's that contrast. You figure out which terms go in the numerator and the denominator by that by hand algebra, but we can look and see roughly what's going on. We're comparing treated versus untreated for P402, and then we're doing the inverse because it's one ratio, P402 on top, over another ratio over ratio, just like simplify that to multiply by the inverse, and you get control versus treated for P401 in that numerator versus denominator comparison. Run that. Here's your results. Plot the MA plot, very similar to the direct comparison of treated P402 to treated P401 because the controls were relatively similar. But now we've got that little bit extra bit of, well, control in the comparison. We can be more sure that the results are what we think they are. All right. Closing in on the, oh, do we have, nope, I just closed out the presenter viewer. Go back to presenter viewer. 2 more sections. Hang in there. I know it's been a while. Hopefully it's been at least informative. This is possibly my favorite section for whatever that's worth. Shrinkage correction and calling significant features. This will get to that question previously about those NAs and the adjusted p-values. I almost never use the default adjusted p-values. And instead, I use post-hoc shrinkage. So the overall goal here is, as with any different statistical testing methodology, is to, of course, maximize power. You want to call as many true positives as possible, meaning minimize the proportion of false negatives. But you can't do, or at least you probably shouldn't do this the naive way by just increasing your threshold of significance. If you call everything above p-value 0.5 significant, you're probably going to call all of the true positives, but you're going to get a whole bunch of false positives in there as well. So you need to control for something. For this type of sequencing-based comparison, we're typically controlling on false discovery rate, FDR, and that is looking at the per comparison. Often this is framed in a Bayesian context, so I don't have to do the usual hand-wavy, careful words of what a p-value technically means. Now, an FDR usually is defined the way you think it means. It's the probability that a significant call is actually follows the null hypothesis, because we're in a Bayesian framework. Hooray, Bayesians. Usually we control on FDR 0.05. I like 0.01 for reference, or even 0.001 if I'm feeling stringent. Those are the overall goals. It would also be nice, and I flag this because we're going to do it, be nice to improve interpretability and reliability of those results. 2 notes for this. Traditional methods for controlling FDR, because they have to be flexible to however misbehaved the data might be within limits. There are assumptions to the testing procedures, but the assumptions tend to be pretty weak. And so within those weak assumptions, the FDR method has to be valid. And that means it tends to be conservative. You're going to have more false negatives than you might like with typical FDR methods. And the second note is, and this is how we're going to create a shrinkage method, is we can leverage extra information to boost the power. We can look at not just the p-values, but say the test statistics and the standard errors together, and possibly some other information. So a little bit of theory here. This has not been used as much, I think, in the literature, although it's fantastic work. John Story, 2003, defining the Q value. I believe this is the first paper where he did that. Q value is formally defined by this complex statistical formulation or probabilistic formulation. It's not as nasty as it looks. This is just saying For a given test statistic t, define the q value as roughly interpreted as a different kind of adjusted p-value. Define it as the probability that the null hypothesis is true, given that your t is in the rejection region, so you're calling it significant, probably that the true coefficient is 0, even though you're calling it significant, and find the smallest rejection region where that's true. That will become more clear in the last line. This relates in the same paper, it defines instead of just false discovery rate, a local false discovery rate. For the standard null hypothesis that your coefficient beta j is 0. The local false discovery rate is almost the same as the FDR. The probability that your true coefficient is actually 0, null hypothesis is true. But now it's not conditional on just your p-value. That's standard FDR, at least for most correction methods. Now it's conditional on your observed effect size, your observed standard error, SJ. This is not size factor SJ, this is standard error SJ in this notation. And new value pi hat. This is an aggregated value calculated from all your tests. This is one more way we can leverage extra information. And this is an estimate of the overall proportion of tests that are going to follow the null hypothesis. And so with this local false discovery rate, essentially just standard false discovery rate for comparison J, but now taking into account all the information from all your tests simultaneously. That's another way to read this line. We can give a much simpler definition of the Q value than the above formal statistics, which is for a given coefficient, and we're going to order them from most significant by LFDR to least significant. Sum up all of the FDRs that are smaller than FDRJ, take their average, and that is your Q value. So this has a very clean interpretation. This says if you say reject or call significant any coefficient or any test that has a Q value of, say, 0.05 or below, then your expected proportion of false discoveries is going to be exactly 0.05. Perhaps something confusing, this is not actually true if you take just an FDR corrected p-value. If you threshold just FDR corrected p-values on, say, 0.05, then you expect your total proportion of false discoveries usually to be less than 0.05, probably by a lot. And that's because most of your significant calls have FDRs that are very small. It's only your least significant result that has that FDR of 0.05. And so your average false discovery rate, if you threshold FDR at 0.05, is going to be less than 0.05 for typical data sets. This is more useful, generally speaking. This means, in my estimation, what you think it means. But we're not going to stop here. We're instead going to look at not the Q value, but the S value from possibly my favorite paper. Again, read into that whatever you like. Matthew Stevens, 2017. He defines the local false sign rate. This is, despite the even grosser, perhaps, formal probability definition here, this is actually a very simple interpretation of what's going on. We have local false discovery rate from the last slide. Local false sign rate then is just what it sounds like. You've calculated a coefficient. It has some value, and that value has some sign. Maybe the coefficient is positive, meaning positive response, a negative response. Vanishingly small possibility, but maybe you calculated a 0 response. And your local false sign rate is going to be the probability that the true sign of the true coefficient, which you didn't observe, you just got to observe the coefficient as influenced by the data. So what is the probability that the true coefficient has a sign that's different from the sign that you calculated from the data? And this is influenced by the Tukey quote, which I've listed below. All we know about the world teaches us that for the effects of A and B, they are always different in some decimal place for any A and B. This, at least to me, is very intuitive. Whenever you're comparing two things, they're always different. They may not be different to a degree that is practically meaningful to you. That's the in some decimal place bit. They may only be very slightly different, but they are different. And if you believe that, then that would suggest inherently that there is no such thing as a false discovery in the formal definition of false discovery rate, because all your effect sizes are non-zero. Some of them may be very, very small, but they're all non-zero. This is a bit better. This is addressing the question, which I think is more practically oriented, which is that usually we care not just is there an effect overall, zero versus non-zero, but are we calculating the effect direction correctly? If we say that a gene is upregulated based on the data, is it actually being upregulated in your treated versus untreated condition? And that's the false sign rate is going to give you. If you threshold at a false sign rate, or the S values we'll get to in a second, then you expect that that proportion of your calls will actually have effects in the opposite direction than what you thought they were. So the S value follows directly from the LFSR, just as the Q value follows from the LFDR. It's the average proportion of false sign calls, analogous to total proportion of false discoveries, among everything that you call significant. And this has one more nice thing, that is just because of the nature of these statistics, the local false sign rate and the S value are a little bit more robust than the local false discovery rate. They require less stringent assumptions on the data. And so this tends to be more powerful than LFDR, meaning that while still controlling the local false sign rate at whatever threshold you think you're controlling it at, your power tends to be larger. Your proportion of true positives to false negatives is going to be higher than the same kind of threshold in a LFDR framework. If you don't care about local false sign rate, then you can use a Q value instead, but I am a big fan, obviously, of the S value. Not going to get too much into this slide. This is looking at the high levels of the ASH model, adaptive shrinkage, by Matthew Stevens. This is how we actually apply and implement S value correction. There's other values, but this is the one I'm going to be showing you in my favorite, right now at least. But I want to mention a little bit, because even though the section is called shrinkage, and indeed the method is called adaptive shrinkage, so far I've just been talking about Q values and S values, and there's been no shrinkage to be seen. So let's look at how this is done. First, suppose we have our true coefficients. We don't get to see them, but it's whatever the true effect size is, beta vector, beta 1 to beta j for all our coefficients, and what we actually get to see from the data, beta hat to beta j, with corresponding standard errors, S vector, S1 hat to Sj hat. This is the core bit here. Hypothesis testing based on these input values, beta hat and S hat, is based on this particular probability relationship. That is to say, the probability distribution for the true value beta, conditional on what you actually got to see, the empirical beta values and the standard errors, that is proportional to some model of beta based on S hat, and some model of beta hat based on the true beta and the S hat values. The guts of that here are here. It reduces to a, in the most typical implementation, a mixture model of normals or a mixture model of uniform random variables. Those details are not too important for us. The main point is that this is constructing a Bayesian model on both, it's constructing a Bayesian model specifically on the underlying and unobserved values, variables, because it's Bayesian, beta. Not beta hat, the model of the underlying betas themselves. And so from that Bayesian model, we can do two things. We can calculate directly from that model the probability that the true value beta, well, rather the sign of the true value beta, is different from the value of the corresponding beta hat. That's your LFSR, which we can then convert to an S value. But we can also, because this is a Bayesian model, calculate the maximum a posteriori prediction map of our beta values. And the particular way that this model is built means that all those map estimates are going to get pushed towards zero. That's your shrinkage. And so this is nice, I would argue, because not only are we using the S values to call significance, which are robust for the reasons I've suggested, but also we can say in a more conservative way that if we've called a particular coefficient significant, then we are confident not only that the sign is correct, but we're pretty confident that the effect size is at least as large as the effect size that we're reporting. This is particularly useful in scenarios like that ratio of ratios model I was showing before, ratio of ratios testing scenario I was showing before, or in fact, just a ratio scenario in general. It is not uncommon for RNA-seq in general, and especially the sparser stuff like single-cell RNA-seq, that for some set of genes, your observed expression levels for all samples in some condition are zero. That is, for some gene, you've got zeros for all of your replicates within a condition, and then you want to compare, say, your treatment group, which has upregulation positive values, against your control group, which has all zeros. How do you calculate a ratio of something over zero? You can't. But what this does is, first off, DESeq will build coefficients with a stopping criterion. Don't just let all zero values converge to a coefficient of negative infinity. Just cut it off. But it'll give you, based on the dispersion parameters, a standard error on that. And then we can shrink that ratio down to a sane level. We'll never know for sure what that true ratio is going to be because we never got a meaningful estimate of the mean on the data that was all zero. But with this kind of shrinkage, we can say with some degree of confidence that whatever the true ratio is, it's at least as large as the shrunken beta coefficient. So that's another reason to use these. More practical one. All right, let's look at this in practice. All right, shrinkage correction section. Very easy to apply. Let's just apply it to the full DDS model. This is using both treated, untreated condition and single versus paired in model for the Bacilla data. Again, comparing treated versus untreated. But now instead of calling results, we're going to call LFC shrink on that model. It accepts the same contrast format if you use type equals Asher. That's the Ash model that I was just mentioning from Matthew Stevens. If you're going to follow my advice, always have S value equals true. That will calculate as your adjusted test statistic. It will calculate the S value. It does not, for some reason, output by default. And we can view those results. Here's your base mean, that's unchanged. Your log2 fold changes now are your shrunken log2 fold changes, standard errors, and now your S values, which is my preferred metric for significance for this kind of comparison. And we can look at that exact kind of zeros scenario where we have all zeros across all replicates for some set of genes, find the zero genes in one group, pull off the shrunken results. The top ones for those zero genes, just visualize the count data to make sure that they are in fact zero genes. And here they are. These are genes where in our treated group, we've got all zeros in the raw input data, but nonetheless, we were able to pull off meaningful estimated log2 fold changes. These ones happen to have completely non-significant S values, so you would never report them. But this is showing the kind of idea of how this works. And if you pulled a gene that, say, had untreated counts not on the order of 1, 2, and 3, but 10, 20, and 30, that would probably return as significant. There is another way that you can do shrinkage correction in DESeq 2. It's a good method, but I'm actually going to suggest against it, and that's ApeGLM. This formulation does not, unfortunately, accept the full contrast comparisons. We have to work just with the one condition model and look at the particular coefficient we're interested in. So untreated versus treated. View these results. Here they are. This is possibly the biggest reason, though, that I am not going to recommend ApeGLM. I'm not quite sure why it does what I'm about to show, but let's switch the order of coefficients around. Let's look at the metadata, re-level to make untreated the default condition, build a new model with the re-level, plot out results names. Now we're looking at, instead of untreated versus treated, we're looking at treated versus untreated. That's the switch we did. Build the ApeGLM based LFC shrink and print out that one. And just for recollection, let's print out the untreated versus treated instead of treated versus untreated. Pull up those results, and this is the issue. All we did was switch the order of which coefficient is modeled as the default, but our log to fold changes have not switched by just a sign. They've also switched in value. And maybe this is fine, but I do not have a good enough explanation for why this is happening to fully trust it. Maybe we can get hold of Mike Love sometime and see if this makes sense. But I did just want to point that out if you see or decide to use ApeGLM yourself, that it has this non-symmetry characteristic to it. That said, we can look at just the S values of both methods and see that broadly they're comparable. Notice the negative log 10 scales for the S values. Larger is more significant, and as the ApeGLM S value goes up, so too does the Asher S value. So if you use ApeGLM or someone else uses ApeGLM, your results are going to be directionally correct. I would not worry excessively about it, especially since we're probably doing post-hoc, say, gene set enrichment analysis, something aggregation based like that, where directionality is going to be more important than specific results on one single gene. All right. So that's a natural break point in case there are any other questions going right now, because we're about to switch over to a quick single cell RNA-seq analysis pipeline. If you're not feeling like posting a question right now, no worries. Think about them if you have any, because we're going to be dealing with a larger data set. There's going to be some more compute time, and so there's going to be a bit of dead air potentially in that example. So just broadly, some considerations for single cell, and then one particular special case that I'm going to show in the example. First, I've hinted at this before in the normalization section, the sparsity of single cell or spatial or whatever other sparse data is going to cause normalization problems. Remember that median ratio, the default in DESeq 2, uses that geometric mean in the denominator. If there's a 0 anywhere in a gene, the geometric mean doesn't exist. And so the median ratio method will fail. Use SCRAN instead, is my current recommendation there. I have a quick question. How is the chance that direction of change is wrong, different FSR from the chance that there is no difference is wrong FDR? Is there an example or a reference we need in more detail? The chance of direction of change. No worries, Jamie. Let me just parse the meaning of your question for a second. Yes. Ah. Um, so it's going to be a little technical, unfortunately, but I would recommend reading that Matthew Stevens paper that I reference in the method in the section on that method, not necessarily for the gory details of how Ash is implemented and how it's interpreted, but in the last section, I believe it is, he has several sections on both simulated and I think some real data where he looks at comparing, say, S values versus other methods of statistical control. And so that might get to the sort of the practical examples of how these various methods compare. Um, as for the interpretation of FSR versus FDR, they're going to be, in one sense, a little bit uncomparable because you're comparing different things. I am suggesting that what you care more about is an incorrect sign call, but that is a much different null hypothesis than this typical FDR. Typical FDR, probably more familiar with, that is null hypothesis. The true coefficient, the true effect size is 0, which is there's no effect. With false sign rate, if you observed, say, a positive effect size, then the null hypothesis is that the true effect size is 0 or anything negative. And anything negative is a heck of a lot of possibilities. This is related to why FSR is more powerful. I think the takeaway I would suggest is twofold, maybe unsatisfying because I'm not sure I'm going to directly address your question, but I would suggest first, think about which type of error you care more about. In my analysis, I usually care more about getting the sign correct than 0 versus non-zero. It's a little bit more stringent, actually. Um, and uh, similarly, think about, I guess, how much power you want to extract out of the data, because FSR does pull more power out of these comparisons. Perhaps a bit of a unsatisfying answer to the question, but they are not quite as comparable as you might like. That said, one thing you can definitely do very easy is just calculate the Q value. There is literally an R package called Q value. I think it was written by John Story. Um, all you have to do is throw in, I think you can just throw in your vector of P values. It's not best when you just throw P values at it, but you can just take the literal DESeq P values, throw it into Q value, and it'll convert them into a Q value for you. Then secondarily, calculate your S values from LFC shrink and just empirically compare the overlap between the two methods. And that might be a reasonable just sort of QC check because they should be pretty comparable between the two. They're both robust methods of false either sign or false discovery control. And if they do diverge significantly, then it might be worth checking why. Little follow-up, let's check. Pair interpretation. Sounds good. Good luck. All right. Consideration single cell. Scran for normalization, just mentioned that. Data sparsity, whoa, don't move that. Data sparsity affects lower bounds. Yes, unfitted dispersion parameters. By default, DESeq does have a minimum threshold. I think it's 0.5. If the fitted mean is less than 0.5, this might have caused some of our problems in the original RPM example. If your fitted mean is less than the default threshold, it won't fit a dispersion parameter. But for very sparse data, most of our gene means are going to be less than that default 0.5. So the recommendation is to set it at 1e-6, 1 in a million. You're probably looking at cells that are non-zero at least 1 in a million times, at least for the genes that you care about. Min replicates for replace. Yes, this is important. You want to set this to infinity. much higher than the default. This is essentially preventing the interior checks to correct outliers. Here for single cell data, we expect certainly the possibility of greater variability, but also in particular more heterogeneous cell types that might otherwise look to the model like outliers, but are in fact not outliers. They're just rare and unique cell types. So this is just turning off outlier correction. Use at your own risk, but I haven't had issues with it. And finally, use T, set that to true. That's just boosting ever so slightly the essentially the standard error, roughly speaking, on the testing to account for some of that extra sparsity and heterogeneity that you might see at a single cell data set compared to the bulk data sets DESeq was originally built for. All right, let's just clear the environment and switch over to the single cell pipeline. Let's see, load up all our libraries. If you're following along and you didn't load this one, do install sparse matrix stats. That's the package I mentioned at the top that wasn't included in the original email. Apologies for that. Let's load the data. We're going to pull some of the, at this point, somewhat classic PBMC 68K data from 10x. If this is the data set I believe it is, this is sort of like the original 10x single cell data set when they published their protocol for the Chromium controller. We're going to downsample that because I don't want to waste your time analyzing 68,000 cells. We'll merely sample a random set of 5,583 cells of this data set. Make sure that the data in PBMC 68K is formatted as a sparse matrix. We can view that right here. Sparse matrices are nice when they're supported because they're smaller, and these data sets tend to be very large. Sparse because they don't explicitly store all the zeros. That's all these dots. The dots represent entries in the counts matrix where no counts were observed, but a zero is not explicitly being stored in the code. It's just implicitly assumed to be zero. So it takes up much smaller space and calculations can be run on it much more quickly typically. So make sure it's sparse. Check the original dimensions of this data. We've got, oops, I did that earlier. We start with almost 33,000 genes, do a quick filtering on this. It's a bit crude, but these data are very sparse. We're looking at the number of non-zeros per gene, row sums of the counts that are greater than zero. So the number of non-zeros per gene and selecting just those genes where at least 1 in 100 cells are non-zero. So if you've got a rare cell type that you want to identify, fix a threshold like this appropriately. If you think your rare cell type is 1 in 100 cells, then pick a 1 in 100. If you think it's 1 in 1,000, maybe 1 in 1,000, or a little bit even smaller, but something to make the data a bit more sane than 33,000 genes, most of which are zero. View the matrix. We're going to be running this in parallel. This one gets set. Yes, I did get set. We'll be running with 6 cores, so it doesn't take actually forever. Just run this. It'll take a moment. This is where we calculate the normalization size factors. I mentioned we can't rely on DESeq defaults here. They will crash or do something unexpected because of the sparsity of the data. So we're going to use Scran. Aaron Lunn, the author maintainer of that, has been transferring a bunch of this code to Scuttle. So don't actually, for the purposes of normalization, don't load the Scran package anymore. Scuttle is implementing the same method, and this is the currently maintained package. The relevant function is compute pooled factors on our PBMC 68K data. This is the function that's formatted for let's just look at this one. This is the one that's formatted for a single cell experiment data structure, which contains our count data internally. If you are using a raw counts matrix, sparse or otherwise, then use the pooled size factors method instead. That can take just a basic matrix instead of the compute pooled factors, which can operate on a single cell experiment. Then we're going to, just so we have it for dimension reduction and clustering, we're going to calculate the log normalized counts. That's just divide each column of your counts matrix by the scran-based normalization factor, add a +1 pseudo count by default. Let's look at log norm counts. There's your pseudo count of 1 by default, and then take the log of all that. Just a log 1p scaled normalization. We won't be using this for testing. See my previous conversation about GLMs, but we will use it for this section. So we'll first pull off some variable genes, do PCA dimension reduction to 20 dimensions on those variable genes, and then run UMAP just for visualization purposes on that data. Here we are. Here's our 5,000 odd cells in the UMAP projection. We can see different cell types. For our purposes today, I'm not going to bother annotating them, but I will cluster them. This is using the scran simple cluster cells method based on the PCA, not the UMAP. Annotate each cluster by a letter instead of a number, just for visualization, and then re-visualize the UMAP colored by cluster. There we go. There's our different cell clusters, if not necessarily cell types. And now let's build a model to, let's build a model to consider each different cell cluster as a different condition, if preparing for the scenario where you want to compare, say, the gene expression of one cluster to another. I'm using, again, the manual size factors right here. Well, that's chugging away. We'll look at some of the terms here. One thing we're doing, use T2 min mu, parallel true, min replicates for a place. We've already kind of gone through all of these. One thing I will note, we're not doing it today for two reasons. One, this GLM gamma Poisson term for fit type. You need a separate package installed to run it, and you can only use the likelihood ratio test, which doesn't support effect size calculation or shrinkage. That's why I'm using the default walled test. But if you don't need effect sizes, all you want to do is find your differentially expressed genes, just full stop. You don't care about what direction the differential expression is or how large the differential expression is, just you want your DE genes. The likelihood ratio test is better generally for single cell data, and the GM gamma Poisson method for calculating your dispersion parameters is much faster than what we're going through right now. And so those are alternates that you might consider as this chugs away. Good moment, and we'll have a couple more of these before the end for any lingering questions or new questions. One note on compute time, and we will see this a little bit later, is it's not just a function of your dataset size, which is 5,000 cells in our particular case and about 6,000 genes. It's also a function of the complexity of your experimental design. And so we're looking at, I forget, on the order of a dozen different levels for our cluster covariate. If you reduce that, it will run faster. It has less stuff to estimate. To the question, I'm just going to get this running because it'll take a moment as well. we're going to compare among the cluster covariate group A to group B, LFC shrink, S value true. Just visualize that quickly. And then let's see, we have the question from Elizabeth. You've been subsetting the data matrix before running DE for both bulk and single cell. Do you recommend this? Or just, yes, I do recommend this. This is not just decreasing the dataset size for demonstration purposes. I mean, it's a little bit that for our current analysis because it's big data comparatively, and it takes a while. But that's more the subsetting to 5,000 cells than subsetting the genes. The reason I would suggest to subset the genes is based on sort of some assumptions on both what you want to test and how you want to maximize power. Roughly speaking, the fewer things you test, the more power you're going to have on the remaining stuff that you do test. And now we get into the question of what are your baseline assumptions? And that's based on your assumptions of both what is a true difference as well as what is a difference that you care about. Separate but related questions. The particular thresholds for how you filter genes will depend on your answers to those questions, but I have been implicitly supposing that if gene expression in at least some condition, doesn't have to be all of them, but at least some condition is not above, say, 1, 2, 5, 10 counts, then either that's something that I a priori believe is not going to be significant, so let's throw it out to improve power, or maybe more justifiably, that's a difference that I don't care about. That gene is so lowly expressed that even if it does return as significantly different, it's not a difference that I care about. Obviously, think carefully about whether that assumption applies to your data. There will be scenarios where you have lowly expressed genes, but those genes have high impact on the cells that they're regulating, and so that kind of filtering would not be appropriate in that case. Apply this on a case-by-case basis. All right, S values calculated and ran. A versus B, this is just showing that we can do it. Although let's look at some of the model characteristics. Here's our dispersion estimates, plot dispests. Looks roughly fine, a little bit sparser per se. This is not the kind of sparsity I was talking about, but visually a little sparser than, say, our bulk analysis, but nothing crazy. But this is a bit odd. This is our MA plot for this particular comparison, and we see a couple things. First off, we have these truly massive fold changes. This is not just 20. Remember, this is log 2 scale, so it's 2 to the power of 20 is the estimated fold change. I'm not sure I believe that ratio, and this is after shrinkage. Also, we have so much stuff calling it significant, which is so close to an estimated effect size of 0 log 2 scale. Almost, not quite, but almost 1/3 of our tested genes are calling it significant. And even for very different cells, I would be very surprised if 1/3 of our genes are, in fact, differentially expressed, at least at biologically meaningful levels. One issue here is not something we're going to be able to get around. One issue is just you have lots of cells, and so the model sees lots of cells as lots of replicates. And in one sense, it's doing what it's supposed to there. You have lots of replicates, you increase your power to identify real differences, even if those differences are small. So you might want to also go in and not just threshold on, say, S value, but threshold on a minimum effect size. But let's see if we can find another reason why this might be happening. And first, let's do like we did with the ZBTV3 example with the ChIPseq and split out our data into cluster A and cluster B and build models just on those. Let's block this, let it run while I point out what we're doing. So it's the same kind of setup, but now we're subsetting our columns for the graph clusters that are A, metadata rows A, size factors A, and likewise for B, and then otherwise just fitting the model identically. And then once that's all done, let's spit out the dispersion estimates. So as this is running one of the We have a question from Will. Can we comment on why you prefer this scRNA-seq method over using Seurat's find markers function or pseudobulking, then using DE-seq to pseudobulk counts? Yes, and I am fine with either method. In fact, if you are actually interested in pulling high variance genes, Seurat's find markers, it's probably better. For the purposes of today, I didn't want to add yet another package and yet another data conversion method. I don't really like Seurat's built-in differential expression testing methods. So you'd have to convert from a Seurat object back to counts or back to single cell experiment to run that. That's why I'm not using Seurat's find markers, but I do like that method if you need to find markers. As for pseudobulk, that is a little bit more nuanced. Certainly that can remediate some of the issues I've just been pointing out for sure. You do run both the added complexity of how do you implement that pseudobulking pipeline. And for anyone who's listening who doesn't know what I mean when I say pseudobulking, that is to say, take your groups, however you define them, and pool all the cells together. Literally sum all the cells in a group you want to compare into one pseudobulk sample, and now run DE on these pooled samples, which of course have a much reduced column space. The challenge there is one more of pipelining and interpretation, and that is, how do you do your pseudobulking? And there are better and worse ways. The obvious simple way we could do here is just to pool within cluster, but now you've got the question of how much should we trust the clusters, especially where a cell is on the boundary between one cluster and another cluster. And I made the decision both for the case of simplicity, but also for the case of calculating variance, that we would like to preserve the excess variance from the heterogeneity of all the cells within the non-bulked group, where that variance would get collapsed if you add them all together. This is all to say your dispersion parameters are going to get pushed up if you don't pseudobulk. So times 2 pseudobulk, it can give better results, but you want to be careful that you're sure about why you're pseudobulking. Why won't zero inflation affect the estimates? Great question. Really good question. Let me get back to it once I'm running another chunk of code that's going to take a moment. Let's look at this here. So not nearly as dramatic as the RNA ChIPseq multimodal comparison that I showed you, but nonetheless, compare the all cluster model, which had groups A, B, and all the other groups out to G or whatever we had, compared to cluster A only and cluster B only. And for comparison, I've plotted in green the fitted curve from the all cluster model. And we can see that uniformly in the case of cluster A, and at least for all the non-super lowly expressed genes in cluster B, that the fitted dispersion parameters are higher than the global group, suggesting, as with that previous scenario, that our fitted dispersion parameters for the global comparison are too low. That for at least this comparison, A versus B, we are modeling the variance as too small with the increased false positives that implies. So whether this is fixable is going to depend a little bit on your pipeline and how long you want it to run and how convoluted you want it to be. But if we're sure that we just want to compare A and B, or generally do pairwise comparisons, one thing we can do is just rebuild the model on just the data that we want to compare. So let's subset our genes now based, that's going back to my previous comment about how we subset genes. Pick your assumptions, but I'm going to say we want either there to be 5% non-zeros for a gene to stay in count A, or 5% in group B, or combined 2.5% non-zero in both A and B. That's this chunk of logic here to keep genes. We're throwing out a bit over a third of the genes that are just so lowly expressed that they don't meet that threshold. And now, first thing we're going to do is take that original merged DDS, subset the genes, test that, let that run. We got a question about zero inflation on hurdle models. It won't affect the estimates. It won't affect the estimates at all. Why do I say that? Because these data are not zero inflated. I have looked at a whole bunch of UMI-based single cell, and that UMI-based might be important because of the deduplication that happens when scanning the UMIs. But at least from that deduplication, when you look at the data after quantification, yes, there are lots of zeros, but time after time, dataset after dataset, I and others have looked, there are not more zeros than you would expect from an appropriately fitted negative binomial or even Poisson model, which is to say there's no zero inflation. Yes, a lot of zeros, but the expected proportion of zeros. And so hurdle models are completely unnecessary for this data type. If you are looking at an older, say, plate-based non-UMI single cell data set, you might want to check for zero inflation. There's no duplication there, obviously. You might have actual zero inflation, and you might want to use a hurdle model there. But 10x style, I've never seen it. Shrunk A versus B subset table out here. Let's look at the plot results. So that's helped a little bit. If we pull that over, blow it up, certainly we've gotten rid of some of this low tail noise where we just have massively inflated fold change because it's all zeros in one group and a couple non-zeros in the other. And you've got lots of apparent power because of large sample sizes, but low counts and ratios over zero. So it's somewhat nonsensical calculated fold changes. But even in the core here, there's less positive calls, but it's also less data to call on. So it's not hugely better. So let's go and do what we did with the ZBTB data. And let's just grab the whole chunk. Yep. All right. And this will not always be an option that's available, obviously, or it might not be one that you want to bother implementing. But in a case like this, where we're getting results that I frankly do not believe, one option is to literally just subset the data to be both the subset of genes, if you believe that that subsetting criterion is reasonable, and also subset on just the cells in group A and group B, and then refit the data to that targeted portion of the data, and then run everything. It'll take a moment, less time because there's less data and fewer comparisons, but a moment nonetheless. All right, that spit out. First thing, many fewer by proportion true positives. That's encouraging. Let's look at our dispersion fits, the original main result versus the new targeted. And the y-axes are mostly the same. And we can see, yes, as expected from that previous analysis, our targeted data set dispersion parameters have increased uniformly. They're almost like a factor of 10 higher. So that's encouraging. Now let's look at the testing outputs. And we have three different groups. We have our original model just with some gene subsetting, and now we have our more targeted analysis. I'm still not sure I believe this stuff, or that's out at 20 plus and minus. Treat with the caution. You could also pseudo bulk if you believe that pseudo bulking is appropriate for your data. That would definitely help there. But the stuff that's closer in, now we've got much less of it. I trust it a lot more. And we're pulling even out of the highest expressing genes, significant fold changes that are also meaningfully non-zero. So if that's available to you and you're getting these kinds of results, single cell is a bit of a challenge broadly. This might be an approach that's worth exploring. And with that, I'm at the end of the demonstration. I'm at the end of the slides, and we're just about out of time. I think, Will, you wanted the last 10 minutes for some wrap-up stuff. So unless there's any final questions, I'm going to turn it over.
Yeah, let's give maybe everyone about 1 or 2 minutes just in case there's any questions, and then we'll go ahead and hand it over.
We got a question over when to use pseudobulk and when not. So from the pure statistics point of view, generally pseudobulking is going to help with some of these issues with, let's say, the massive, unbelievable fold changes, and also some of this excessive power out at the higher expressed genes, because you're just going to pool all those samples together. DESeq is going to see that massive reduction in N, in number of samples. And so your standard errors are going to appropriately increase, and you're going to get less significance on your p-values and more rational effect sizes. That's all good. Challenges are, in our particular case here, where we had, say, just the one data set and the different clusters. Let's say I pooled all the cells in one cluster together, and likewise for all the other clusters. Now I've got, say, 12 clusters. Now I've got 12 samples, each with a replication number of 1. It's going to be very hard to calculate dispersion parameters where every condition has one replicate. You can do funny things like build a model which is intercept only first to calculate dispersion parameters, and then save those dispersion parameters and rebuild the model with all your covariates back and test them. So there are hacks around it, but that's a practical issue. And then there's also the question of how well defined are your different groups. If it's purely computational clusters, then you want to be reasonably confident that the cells allocated to one group are actually different from the cells in the other group, because you're going to be pooling and thereby shrinking the internal variance within cluster by pseudobulking. And so that's going to be more of an interpretation constraint. Hopefully that helps.